Yes, so I would talk about biological intelligence and how we at BrainCures apply it to facilitate precision medicine. So I think not many people have heard about biological intelligence. So the definition is that it's a biology and data-powered approach that eliminates preclinical failure and enables at least 60% clinical trial success rates. And we do that all without any deep learning steps associated with artificial intelligence, AI. This is probably the only time I will use AI because what we will be talking is about how a biology-first approach enables precision medicine. And so now I will play a short video that explains what we do. Hopefully the screen is back on, and so we, as a company, you probably it might be the first time you're hearing about BrainCures. We have been working on building the company since 2017, and we're going from height to height, from different awards at different startup accelerators and then biotech forums. And we've received many positive client comments, and we've recently been featured in the Longevity Technology magazine in an article called Move Over AI, Here Comes Biological Intelligence. It's a very nice article. I encourage everyone to see it, and I will explain what the short video told you is we have developed how we go beyond machine learning techniques to enable success. So just because I don't know, the audience may be general or not be aware, in neurology we have a very big problem with the clinical success rates. They're essentially preclinical success rates are less than 2%, and it's just a very competitive area. And the reason for that has been poor biology. And when I talk about brain diseases, it's important to say that I'm considering different ADHD, so attention deficit disorder, Alzheimer's disease, depression, epilepsy, pain, Parkinson's disease, schizophrenia. And then at least 1 in 10 people are affected by brain disease. And we have an aging population. We have, so today's industry average is a 1 in 10 success rate, clinical success rate. In Alzheimer's disease, that's a 1 in 25. And this is a long list of brain disease that BrainCures says that we target. And the reason why we can target is that we have our biology is focused on synaptic plasticity, long-term potentiation, which is just a you could say neurologist term for how memory is formed. And I think a key thing to anyone considering the neuro space is that it's a growing market. In 2019, neuro drugs generated over 80 billion revenues. those revenues will be are projected to exceed 131 billion, and Alzheimer's disease will be a key one there because of just the aging population. So how can we overcome and increase success rates? Most people may be aware of AstraZeneca's 5R framework, where they have looked and shown that essentially if you don't have a project that's guided by biology, you will fail. And biology means that you just understand what the target hits and the genetics of the population, and possibly having a biomarker to stratify your patients. So that's needed to generate, what's needed now is actually better biology based on quality data, and also looking at the transitions that are required. So temporal of how a disease develops or how a disease is reversed. And BrainCures has been so unique that we have focused on development of a physiological process rather than seeing, well, let's take a diseased animal and then go backwards, which is what a lot of approaches have been doing so far. So it's a really interesting approach that we use. And what we've done at BrainCures is it's the biology that powers our discovery engine has been in the making since at least 2004. So over 15 years of science have been put into building the BrainCures Discovery Engine. So we initially started with 3,000 over genes that are responsible for memory formation in a healthy animal, and at different time points. And we then did data mining to restrict that to 822 BrainCures Discovery Engine targets. We then used systems biology approaches such as network analysis, data mining, and proprietary algorithms looking at the different sequence aspects to tease out molecular corporation, where we have identified how these target genes are workers, managers, to directors of brain biology. And as we increase, you could say that go up in the hierarchy, we are able to achieve much higher success rates, and that gives low increasing precision medicine potential as you have a compound that targets a director so as you go from targeting a manager to a director, you increase the success of your clinical program. And I will show some real case studies that prove the point that our algorithm can really drive successful treatments based on biology and biomarkers that we're able to provide. And this is really so the reason why BrainCures is able to, you could say, bypass all artificial intelligence and training steps is because we've taken time to tease out the biology of the brain. And this biology of the brain shortens the drug development cycle by at least 4 years. You can come to us with your question on a compound, disease, or other data. So it could be preclinical or clinical data. And then we are able to, we have at least, you could say, 3 services we can provide. So today we're a consultancy that aims to, with time, to also develop into a virtual pharma company that develops their own, essentially, preclinical to clinical packages. So some of our services include the BrainCures Discovery Engine de-risking. We also, I think, are most unique in having a virtual phenotypic screen that's powered by targets common to the BrainCures Discovery Engine and the L1000 dataset. So you might know the MIT L1000 dataset. That's an in vitro dataset, while the BDE is an in vivo dataset, and we can essentially do a phenotypic screening to provide you with great candidates. And we can also apply, so we have a special way of designating these directors and managers and workers of any process. So if you have a list of genes, we would be able to build you a bespoke discovery engine that would then allow you to do these things even outside of neuro. And we are so we are a small startup, so obviously we are focused on neuro, but we are also very opportunistic and open to partnerships with applying our technology outside of neurology. I think so. I mean, it's a bold claim to say that we can deliver at least 60% clinical success rate. So to prove how we've come to that claim, this is the slide that shows that we have found, so GSK has put together 72 drug target pairs that failed in clinical phase 3. When we use our BRAINCURES Discovery Engine, the hub filter, we eliminated essentially 57 of the phase 3 failures. So if the company had come to us earlier, we would have been able to tell them the 15 phase 3 projects that had some potential to succeed, and prevented the 57 other phase 3 failures, which we understand just one such twist will cost $200 million at least. So, and the success rates are here separated by different target classes. So in some target classes, we can our success or failure elimination rate is 100%. So how can we, you could say, work together? So we have at least 3 paths to success. The first one involves starting with a drug or even gene list to then feed it through the BRAINCURES Discovery Engine and provide you this prioritization that can then be further refined to your context, your disease, through literature searches to then provide you with the pathway to treatments and biomarkers, so personalized treatments. The second path involves starting with, well, you bring us, I think, first you bring us a question, typically saying, well, look, we're interested in such an indication, or understanding what our drugs might target. And in path 2, we can start with drugs for your indication of interest and identify which L1000 molecular signatures it has, and then run those signatures through the BDE 100, which is our bespoke discovery engine. So there's the L1000 has almost 1,000 genes. When we compare the L1000 to the BRAINCURES Discovery Engine, 98 common genes are shared, and that gives us the BDE 100 custom engine. And that gives us the opportunity to in silico, for in silico phenotypic screening that can give you high success rates. And then path 3, you might come with a patient cohort or tell us that you might be interested in a patient strategy, and we'd be able to, by querying so just even the masters, some of them can be assayed readily, and that can give the opportunity for personalized treatment recommendations. So this might be a very busy slide, but the point of this slide is to show how we apply, how the full cycle works in one on one slide. And this here we've started with epilepsy, where the client had asked us to give them some suggestions. And so one starting point is the Open Targets database, where we found 207 drugs, and they were linked to 208 gene pairs. Filtering through our engine, there was 51 of those 208 were targets within our engine, and that gave us 20 drugs. And these drugs were then prioritized by literature-based searches, and we were able to tell the client what their also clinical novelty was. So The higher the score here is, the less, you could say, knowledge there is, so higher intellectual property uniqueness. And then these targets can be prioritized for biomarker pairs, and this client is now working on some prospective experimental R&D to develop epilepsy treatments. And for biomarker screening, there is this, the Genomics England has a panel app, and we were able to start from over 600 epilepsy biomarker genes and reduce them to about 50, and the green ones are the high performance biomarkers already. The red ones are ones that need a lot of evidence. So we have also, obviously, I think most companies today are also taking part in the COVID-19 repositioning race. So we have applied the BRAINCURES engine. Starting from open targets, we found 266 genes linked to coronavirus. We then applied the BRAINCURES engine to find that 31 of those are BDE prioritized, and this has given us 207 phase 3 repositionable COVID-19 drugs. And we'd be happy to discuss this with anyone who's interested in more detail. Another, I think, unique feature of our company is that we've built a knowledge base of more than 2 million articles linked to rare diseases. And this allows us to rare neurological brain diseases. So there's 7,000 rare diseases, and 1,000 of those are neurological. So we are able to start with, say, your compound X structure, and for one of these structures, we were able to identify 70 rare disease opportunities, and that compound from the company was very similar to CDPP, and the company is taking advantage of some of this insight to see how similar or different their drug mechanism might be. So this offers you, you could say, when you, I think, the most optimal stage is to try to use this preclinical to early clinical testing, so you get as many indications as you can in your IP filing. So this is just another example of how we applied our virtual phenotypic screen to Alzheimer's disease. So we started with 128 AD drugs. We then found that only 55 of them had the L1000 molecular signature. And we then analyzed the signatures of the BRAINCURES Discovery Engine to identify negative and positive plasticity modulators. And that gave us 21 80 drugs that have the potential to have a better plasticity profile than the current drugs, memantine and galantamine. And one such drug from this screen is drug X, and it has been shown to slow the progression of Alzheimer's disease in patients with moderately severe impairment in 171 phase 2 patient studies. So this is a drug that has high potential to become one of the drugs that treats Alzheimer's disease, disease-modifying treatment. And today we don't have that for Alzheimer's disease, really. And I think the key point also is that our predictions, for example, for Alzheimer's disease, given at least threefold increase in the likelihood of progression to phase 3. And we calculate that looking at the success rates of drugs linked to 176 AD genes that have, you could say, a clinical success rate associated with them. And we are able to go from essentially the initial list has only 7.5 likelihood of progression to phase 3. When we use our masters, it's nearly tripled. So I think this is a big improvement, and obviously it cuts down also the number of projects that you as a company would need to do to succeed. So I think we're now at a stage of what do we need today. So the company has two verticals. One is the data analytics, and another one is developing our own pipeline. And we intend to do this all through partnerships. And partnership pathways include investments into internal lead development or partner lead development. And I'm privileged to have a very experienced team on board, and that involves myself. I call myself a neuro drug hunter with at least 15 years of experience in drug discovery from both academia and pharma. So I used to work at Lundbeck in Denmark. And then Jean-Francois and Peter Ball, they bring over 45 years of experience in business development and product development. And Professor Jacques Mallet, he's one of our advisors. He's distinguished scientist in neuroscience and gene therapy since 1980s. So I'd be happy to take any questions you may have, and we really look forward to making new connections to allow us to advance precision medicine based on biological intelligence.
Amazing. We have few questions for you. So the first one comes from Mario. How will big data have an impact in approval times and drug development cost?
Well, the key point is it has to be the right big data, has the potential to allow companies to leapfrog most of the preclinical development. I think that's the key point, and many companies are already doing that, a growing number. So the correct big data can significantly allow you to reduce the first 5 years of preclinical drug discovery can be shortened to probably 1 year, where you do a few killer experiments. I think that's the key. I think that's where the key is, and also new biologies can be discovered from the right big data. I think one of the biggest and the most exciting big data that I've seen out there was the Lichud data set of 108 patients, where they were collecting a lot of data. Every 3 months they were testing these people on a metabolomics panel, and they were able to figure out what transitions go from health to disease, and they were able to even tell 3 patients prevent chronic disease from developing. So I hope I answered that for that first question.
Yeah, thank you, Krzysztof.
Right.
Yeah. Another question. Yes, very nice. Another question is, right, it is also a link to this question, it also came from the panel, that how do you see, because currently the regulators are quite rigid, in terms of, let's say preponing the timelines, they are not confident whether, they can omit the preclinical stuff. And also the artificial intelligence scheme of things are not integrated in the regulatory approval process, of NDAs and biological candidates. So what do you think, with this big data, do you predict some transformation also happening in the regulatory agencies like US FDA, EMEA and all that? Is there any cross-border, things which are going on, or are you aware of something which you can share today?
Well, obviously in Europe we have the trustworthy AI stream, where there's a AI H-leg committee got together and gave a lot of, you could say, guidelines for trustworthy AI. The NHS has developed a big, you could say, guide on how to buy AI, because I think when we talk to pharma customers, there's every month there's 10 new data analytics companies that say we use AI to do better drug discovery, and typically that's limited to one phase. And so the companies are very challenged in choosing who to work with. And in the US, obviously, I think the FDA also has developed committees for data analytics. So I think there's increasing regulation or guidelines and there's the Broadcom Commission's report that was made with collaboration with Novartis. That's, I think, a great recent paper showing, you could say, how we can advance or use data analytics. But I think the key point is that data analytics is only as good as the experts that are involved in building it. We've seen some failures. Well, let's talk about the successes, right? So I think one success that's proved how experts are required is the retina scanning of Google DeepMind. It showed that the machine is only as good as the top experts who fed the information there. So the machine will not learn new biology for us unless we give it unless humans use the machine to tease it out. And that's, I think, what we've done with biological intelligence. And that's why we've always, as a team, said we're a biological intelligence company. Biology is what's going to guide our success. And I think there's some great companies like Berk Health and a few others that we know of that have taken, you could say, this approach. So yes,
okay,
I think the impact that this big data can have will be, well, we know on ourselves that it's at least 100-fold if we are engaged from the very beginning of the preclinical drug development cycle. We can give 100-fold saving economy because of the times that you will fail in preclinical, right? Today, the preclinical success rates are at best 20% from going from preclinical. We are able to make that 100%, we know.
Correct. Very good. So thank you again, Krzysztof, for the global audience who are watching, globally today. Dr. Krzysztof Potempa can be reached out on LinkedIn, over LinkedIn. If you wish to contact him, you can also contact us. We will connect to him. But we are all glad that he was here today. I'm sure you all have enjoyed his presentation, from artificial intelligence to biological intelligence, and how to, prepone the process, drug discovery, development, process timelines. Thank you again, Krzysztof, for joining us today. And see you in future Pharmacircuit conferences and also for our mentorship programs. So thank you.